Uh, it's my immense pleasure and honor to introduce to you all and welcome to Living Histories, Professor Roland Merks. Please take it away and tell us about Living Histories.
Well, thank you so much for this really nice invitation to tell about my life so far as a scientist. So yeah, the first picture that you see here is me at the primary school, at an elementary school, at the occasion of the 450th birthday of Leiden University in the Netherlands. And here I'm explaining them about Turing patterns using Legos and cellular automata and pictures of zebras and of zebrafish. And this was really a lot of fun. And what on the next slide that trying to switch to right now, you see me as a boy at elementary school age, and I grew up in a family of five. I was the youngest of five. I had three older sisters and one older brother. My father worked as an elementary school teacher, and my mother worked at home. And I had many interests. So I lived a little bit in my own world, so to say. I loved reading, like Roald Dahl, in Dutch translation here, and I loved playing with Legos and building cities, building worlds, and later on all kinds of cars and machines. I also loved going to the pond and looking at frogs, catching fish, tadpoles, these kind of things. And later on, I had an aquarium, and I played with electronics. And I also had what was called a poetry album. It was a thing at the time mostly for girls. But since my older sisters had one, I really wanted one myself. And a book where you ask friends to write small poems about anything or about you. And this was my primary or the elementary school teacher who wrote a little poem for me. I made this drawing, and I think it was kind of apparent at that time, although I didn't go into chemistry, it was kind of apparent at that time in what direction I was heading. But still, I also many other interests. And in high school, besides being very much interested in the sciences, I was also interested in languages, literature. I had my own literary magazine that you see here in the high school exhibition of. It was a very old high school that is now closed down, called the Dominicus College in Nijmegen. And here you see this literary magazine that I called Carré, that I set up with some schoolmates. And but I also was really passionate biology. By then I had frogs and lizards in my room, and also I joined a really nice place which was called the Jonge Onderzoekers. It means the young investigators, and this was a hobby club where you could play with electronics, where you could also work with computers. This is a kind of old computer running CPM, a popular operating system at the time. And this guy could have been me. I'm not sure. I don't think it's me. But I was very often to be found behind that computer. I learned how to program in assembly, in Z80 assembly, to work with this operating system. And I was really passionate. And at that time, also my parents got a computer at home. This Commodore +4. It was a Commodore home computer that was originally built for small businesses, but later on it didn't work so well, it didn't sell so well, and it was quite affordably at the European market. And I really learned to work with this, all the insides and outs. I wrote computer games and I made my own basic exchange. So it kind of would have made sense that maybe I would have gone to study mathematics or physics, but given my passion for biology and all the animals at in my room, I thought, well, I really want to understand how animals how animals work. And so I thought the best place to do that was to study veterinary medicine in Utrecht University. So there I learned a lot about, I'm sorry, I learned a lot about uh, anatomy of dogs. I learned cell biology, biochemistry quite thoroughly, and I enjoyed it tremendously. Also, there was a local student association called Archaeopteryx Veterinaris, where we exchanged knowledge about veterinary medicine of special animals like lizards, frogs, fish, but also small mammals. And and through that, I also got interested in evolution. There was a library where there was old literature about paleontology, about comparative zoology, and slowly it dared to me that many of my fellow students were not so much interested in all this knowledge that I was absorbing, but they were more interested in becoming a vet. And I thought, well, I'm not sure if I should become a vet. And so I started looking around and came across the study of biology. And in there, in the curriculum, there were two words that attracted me tremendously, and also kind of reignited my love for computers and for math. And those words were theoretical biology and bioinformatics. So I started studying these two courses. First, theoretical biology, and there I met Pauline Hoogweg, who is a bioinformatician and theoretical biologist at Utrecht University. I got completely fascinated by all the things that she had to say, going from cellular automata, differential equations, all the way to pattern recognition. And indeed, I decided to do a master's project with her that was on evolution of development. And also, she recommended I do some work in experimental biology as well. And so that brought me to Tony Durston, where I worked with, not so successfully, but where I worked in the lab on Xanthus lagus, a frog. And he was very much interested also in theoretical biology, and also very much appreciated people working with theory and trying to incorporate theory in biological research. So these two people really put me on the path towards this career. And also at the group of Pauline Hoogweg, I first learned about the cellular pods model, which is a cell-based model developed by James Glaser in Indiana University that I used a lot later on. So that was a really great time, and I worked really hard. And but also then some other things started to happen, because I was working really rediscovering working with computers, and almost worked day and night on this, and my body started protesting. And this almost I was at some moment at the point that I would touch any button and it felt like my arms would fall off. So I was like, well, what should I do? Well, I didn't really know, and I almost decided to find another career. And I'm happy I did not. And one of the things that has certainly contributed to that is that Pauline Hoogweg also recommended me to apply for a grant to study abroad for a year. And she recommended me to go and talk with Professor Koneko, who was earlier in the talks today. And he allowed me to come to his group in Tokyo University, where I really, really learned a lot. So I learned about, I met many of his students, including Chikara Furusawa. I learned about autologous diversification. I learned life in Tokyo. And one of the important things I also discovered was Central, the hot bath culture that definitely helped me, contributed to help me recover from this repetitive strain injury. And I also try to program a little bit with speech recognition, and I'm sure I must have disturbed many of the students at the time, but thinking back of that, they were incredibly tolerant to that. So I still want to thank them very much for that. So after a year, I came back to the Netherlands, also to meet again. So one of the other things is that when I was in Tokyo University, I also met James Glaser. He came and visited Professor Koneke's group and invited me. He was the developer of the cellular pots model that I come to like so much. And he invited me to come give a talk at Sendai, in Sendai, Tohoku University. And this was really a great opportunity. I took the boat to the ship to go to Sendai, an enormous adventure. And I gave a way too long talk. I hope I won't do that today. So then after I came back, I really wanted to go back to the Netherlands because also by then I had met my future husband, Bart, who was still in Amsterdam. And so I was looking for opportunities to do something in morphogenesis back in the Netherlands. And then I read about a PhD opportunity in computational science. And there I met Jaap Kaandorp, Peter Sloot, and Albert Hoekstra, who are computational scientists. And I learned about the Lattice-Boltzmann methods for fluid dynamics. I learned about computational geometry and complex systems. And there I worked on modeling coral growth. And so this is an example of one of the models I developed for my PhD, trying to understand how the interaction between polyps leads them to finally to coral morphogenesis. And also another great opportunity I got at the time was to attend the Complex Systems Summer School at the Santa Fe Institute in New Mexico. So after finishing my PhD, although I liked the corals very much, I really thought back a lot about the cellular pots model. I even tried to build a coral growth model using the cellular pots model unsuccessfully. And then I was first I wanted to do a postdoc. I was first trying to go to New York. I wrote a grant for that, and it failed. And then James Glaser said, well, you could come to Bloomington. I thought, well, I would love to work on the cellular pots model, but Bloomington seemed to be a little bit remote. And I'm really glad I did it in the end, because it was really the best of experiences. It's really a very nice and quiet town where there's so much opportunity for science. So I really appreciated this time. And there I started working on angiogenesis, on blood vessel formation. So one of the things we discovered is that if you have introduced a length constraint in the cellular pots model where you have elongated cells, then that can help understand the formation of blood vessel networks. And I did some experimental work at New York Medical College with Stuart Newman, and we could get some understanding of the formation of these vascular networks. And this has been a topic that I'm still working on. I'm sorry. Yes.
Sorry, you're a little bit over time right now, actually.
Oh, I'm so sorry.
No worries. Just a warning.
I'll just rush over. So after Bloomington, I didn't stay for very long because I wanted to go back to Brussels, to Belgium, because then my husband, Bart, to whom I got married in 2007, was working in Brussels. And but I still wanted to do something in Belgium. And so that's when I met Geert Beemster, who's working in the plant lab. And he really said, I want this kind of skill in the plant lab. So that's when I started to work on plant development. And so this was, again, a completely different topic. But I really appreciate it because it really helped me to kind of moving from field to field really helped me to develop different insights in this topic. Well, I'm sorry that I'm over time. So one thing I really want to say is that after my time in Belgium, I could start my own group, and that was at the CWI in Amsterdam. I received a call that within the Netherlands Consortium for Systems Biology in Amsterdam, they really want to have somebody who would set up a core modeling group of that would coordinate modeling activities of people all over the Netherlands. And this was really an amazing opportunity. So I could start directly by setting up a nice group and could also cherry pick all kinds of interesting problems of the experimental biology groups in the Netherlands working that needed some modeling input. So I could take further my work on plant development, but also I started working on microbial ecosystems. And so indeed, to this day, I'm still working on many of these topics. At a certain moment, I received a phone call by Arjen Doelman. He said, well, you're now working at a research institute. Do you want to come and spend 1 day a week with this at Leiden University? So I'm really happy I did that. I still work on a variety of topics. I also went back a little bit to experimental biology because I felt that at some moment we needed to get a bit more contact with the experimental biology. And well, that's basically where I am now. So what I learned so far, I hope you've seen that I've seen a lot of coincidence. I started studying veterinary medicine, but step by step, I kind of got in touch with using mathematical approaches and computational approaches. I would say, yes, do what interests you most. But if life or other circumstances do not cooperate, find an additional topic that interests you most. And so let coincidence help you a little bit. Also, I worried a lot. At one point, I worried that I should stop my career and do something else. But I think sometimes worrying is productive because that really helped me to find solutions. So with that, I would like to thank my whole research group. There are only a few of them on this picture. And I'm really sorry for being a little bit over time.
Thank you so much for a wonderfully inspiring talk and for these lessons learned that you shared in the end. In the interest of time, I am closing the recording, but I will follow up with you via chat with the question.
Yes,